This is Miss Dagenford, and we want to continue our work in, genetics and meiosis and start with some recapping with the other day. The other day, we were talking about the process of meiosis, and you were left with this problem of matching and sorting the stages of meiosis. And this is what you end up getting. And it's very similar to the diagram that you had earlier. We just rearranged them a bit. One of the key takeaways for meiosis is that the homologous pairs are the most important thing that happens in meiosis. The homologous chromosomes pair up, and then in the first stages of meiosis, the homologous pairs are lining up together in metaphase one. The homologous pairs separate in anaphase one. And then we divide the cell in telophase one to get two cells. But here's the important part, remember, with half the chromosomes. This was a cell that had 6 chromosomes to begin with. At the end of the first division of meiosis, we only have 3 each. Now, the second round of meiosis has to do with splitting the duplicated chromosomes in half. And then that way we have individual chromosomes instead of duplicated chromosomes. But we still only have half the chromosomes. So in each of these daughter cells, we'd have 3 chromosomes, not 6. So important that most textbooks, yours included, usually include charts that compare mitosis and meiosis. And so we can look at that. In mitosis, the goal is to just duplicate the cell, get a copy of the cell. So if we start with diploid, 4 chromosomes in this case, we want 2 cells with 4 chromosomes each. And in metaphase, the individual duplicated chromosomes line up. In meiosis, after we duplicate the chromosomes, the homologs pair up, they create that tetrad, the pairs, and then they line up together at metaphase 1 and then separate from each other, thus reducing the chromosome number. So again, in this case, we started with 4 chromosomes. Each of the daughter cells only now has 2. And then the second stage of meiosis is simply to separate the chromatids to get unduplicated chromosomes. So that really is the key takeaway with meiosis. And you ended with this practice problem, which was just to identify the different stages. And so we're just going to end with our recap with a couple of vocabulary terms. So somatic cells would be body cells that just are performing what they're supposed to do, like skin cells, stomach cells, muscle cells, bone cells. They are our body cells. They are genetically identical except for minor mutations. They start diploid, they have 2 sets of chromosomes, they divide by mitosis, you get 2 diploid cells afterwards. Gametes are the reproductive cells, egg cells and sperm cells. The cells that make them start by being diploid, but then they end up being haploid with 1 set of chromosomes. So what I would like you guys to do is hit pause and see if you can do the sorting activity that describes the difference between haploid and diploid. What would these guys fit? Okay, so if you guys have done this, you can take a look at this. We've got 2 sets of chromosomes versus 1 set of chromosomes. And then diploid would be the result of mitosis versus haploid is the result of meiosis. And so that's just a little sorting activity. That's going to bug me. Let's get them nice and even there. And so that should give you just a little bit more practice. What we want to do now is talk a little bit about the history of genetics as a study. And to do this, you have to go over Gregor Mendel. Gregor Mendel, or Gregor Mendel, I've heard it pronounced both, is kind of considered the father of genetics. And really, he the best part of what he did, he was extremely methodical, extraordinarily patient, and extraordinarily thorough. Those were characteristics that really contributed to his success because it took him many years of painstaking research into the genetics of pea plants to get the results he did. Now, we've got a couple of little videos that you will watch. And as you watch them, you want to look for what he did, the experiments he did with pea plants. One of the successes in his studies is he was highly um, thoughtful about controlling the breeding of the pea plants so that he could track the traits. He looked at several different traits of pea plants to examine whether or not they were passed on and in what way. Part of his success is that he was able to isolate purebred pea plants. Purebred means that you're only going to pass down certain traits. You have no option. You will only pass down those traits to your offspring. Now, one of the things that you want to watch for in the videos is some of the terminology that he used. He did parental crosses. Now, a cross is simply when you're going to breed male and a female together. This applies to pea plants. This applies to dogs and horses and any breeding program that we have. When you refer to a cross, you're looking at, okay, we took a male dog and a female dog and cross them together to get the first generation, or F1. And so F1 refers to the first generation. If you were to then cross those F1s and then you would look at the second generation, that would be similar to grandchildren, if you will. Now, when Mendel was studying his results, he very carefully cataloged the number of traits that he found when he would do crosses. And when he did that, he was able to compare mathematical groups. So it would be very highly useful to us to make sure we understand ratios. And ratios are the ability of making mathematical comparisons. So if you were to compare um, the number of dentists that use Colgate toothpaste with the number of dentists that use Crest or fill in the blank, you could get a number. And then you could make a ratio out of those numbers. Well, 4 to 1 or 1 to, and so that's a ratio when we compare. We use a colon to then juxtapose, put the numbers together. So, for instance, 3/4 and 1/4, you could express that as 3 to 1. Three of something compared to one of something. Same thing with 1/2 and 1/2. You can express it as 1 to 1. Even whole numbers, if you have 50 of pea plants that are tall and 50 pea plants that are short, you could express that as, 1 to 1. It's a 1 to 1 ratio. If I have one pea plant that's tall, I'm going to have another pea plant that's short. And so these ratios, you're going to see them a lot during our numbers. So it's good to practice that. The other thing that we're going to be looking at would be some rules of probability, but we're going to save that for another video. So as you watch the videos, look for those things, and then go ahead and hit pause. You'll watch the videos on Mendel. And then as you watched in the videos, you saw that Mendel came up with a couple of conclusions. He realized that each parent can contribute one factor or trait. He didn't know what that was. You notice in the video, They said that, we didn't know about DNA back in the 1850s, but Mendel realized that each parent provided one factor or one gene through those sex cells. And he called that the law of segregation. So each parent can only give one. Each parent had two, but each parent only gave one to the offspring. The other law he found was law of independent assortment. Now we'll talk about that a little bit next week, but basically it's this idea that if they're on different chromosomes, different genes are going to sort independently of one another. So um, um, if you have type A blood, that does not affect whether or not you're going to get brown eyes from your mom or dad. So those genes, different genes, as long as they're on different chromosomes, they sort independently. Okay, so let's pause for now, and we will pick up with some more practice problems.